So for Chapter 4, Segment 1, it begins with the topic of the entire chapter being the chemistry of behavior, including neurotransmitters and neuropharmacology. The first definition given is what is exogenous, which means anything arising outside of the body. Drugs then are said to affect the nervous system by altering brain chemistry and synaptic transmission. The study of the rest of the chapter is neurochemistry and neuropharmacology, neurochemistry being the branch of neuroscience concerned with the fundamental chemical components and processes of the nervous system, while neuropharmacology or psychopharmacology is a scientific field concerned with the study and discovery of components that selectively affect the function of the nervous system. Both of these are concerned with the processes of the nervous system. Neuropharmacology is more with the exogenous factors, while neurochemistry is more of the endogenous factors. Next section involves the study of synaptic transmission, and it states that it's a complex electrical process. First definition given is receptor subtype, which was discussed in the previous chapter, which just means any type of receptor having functional characteristics that distinguish it from other types of receptors for the same neurotransmitter. These receptor subtypes can either be metabotropic or ionotropic. Metabotropic uses G protein coupled receptors because they're not directly associated with an ion channel. These G protein coupled receptors are a cell surface receptor that, when activated extracellularly, initiates G protein signaling mechanisms inside the cell. So it's a slower process for metabotropic, while ionotropic is associated with a family of genes that code for a variety of protein units that in combination make up the ion channels at the core of the receptors. These are characteristic, there's a characteristic of each type is a unique combination of subunits, and the brain may alter the combination over time of these subunits to alter the function of their synapses. Ionotropic receptors are directly associated with an ion channel, so they're a lot faster, and they're a lot simpler than metabotropic receptors. Neurotransmitters of different receptor subtypes trigger different responses in target cells and are distributed differently in the nervous system. So therefore, we can design drugs that affect only one of the subtypes, thus having a specific effect on the nervous system. In association to what goes into a receptor, that is a ligand, which is a substance that binds to a receptor molecule. Ligands can either be, most ligands are endogenous, they're created by the body, but exogenous factors can either be agonists, have an agonistic effect on the receptors, antagonistic effect, or an inverse agonist. Agonists then initiate the normal effects of the transmitter on that receptor, so they mimic the effects that the ligand has. And the antagonist binds with the receptor and interferes or blocks the action of the transmitter, so it blocks what the ligand would normally do to the receptor. Inverse agonists then bind to receptors and cause it to do the opposite of what the transmitter would normally do. There's different types of ligands that either block receptors, and those can be competitive or non-competitive. Competitive ligands are substances that directly compete with endogenous ligand for the same binding site on a receptor molecule. So when they bind, the endogenous ligand can't bind, and thus they can't have their normal effects. The non-competitive ligand then alters the response of the endogenous ligand without interacting with the endogenous ligand's recognition site. They usually bind to a modulatory site, which is outside of the normal receptor, but it can alter the receptor's response to the transmitter. These can either be agonistic or antagonistic. The next section discusses many chemical neurotransmitters have been identified. Endogenous then, in opposed to what we discussed earlier, is produced inside of the body. There's multiple criteria to be considered a neurotransmitter, 6 in total. The substance must exist in the presynaptic axon terminals. The presynaptic cell must have the appropriate enzyme for synthesizing the substance. The substance released has to be released in a significant quantity with action potentials when action potentials reach terminals. The specialized receptors that recognize the released substance are on the postsynaptic membrane, and through experimental application of the substance, it produces changes in the postsynaptic cells. And lastly, blocking the release prevents presynaptic activity from affecting postsynaptic activity. Then there's multiple different types of neurotransmitters. These are the general categories, which will be discussed in more detail later. The first being amine neurotransmitters, which are based on modifications of a single amino acid. These include acetylcholine, serotonin, and dopamine, are just some of the amine neurotransmitters. And then there's amino acid neurotransmitters, which are itself just an amino acid, which include GABA, glycine, and glutamate. Next are peptide neurotransmitters, which are a short chain of amino acids as opposed to a single one. These include neuropeptide Y, galanin, and VIP, in addition to others. And there's gas neurotransmitters, which are a soluble gas, usually nitric oxide or carbon monoxide, that are produced and released by a neuron to alter another neuron's function. It's hard to prove that one transmitter in one location is a transmitter in another location, though, for all the ones that are discovered, so that kind of leads to the delay in the discovery of some neurotransmitters. Next section is neurotransmitter systems form a complex right in the brain. Brain activity is characterized by a diverse assortment of neurotransmitters distributed in a complex network. Co-localization or co-release is the appearance of more than one transmitter in a presynaptic terminal. The next section is titled Most Abundant Excitatory and Inhibitory Neurotransmitters in the Brain are Amino Acids. And those, as we stated earlier, were glutamate, GABA, glycine, and also aspartate, which is an added one that we didn't discuss earlier. Glutamate is an amino acid transmitter, and it is the most abundant excitatory transmitter. Any cell that uses glutamate as their synaptic transmitter is described as using glutamatergic transmission, and these cells include AMPA cells, kainate, and NMDA receptors. Most of them are ionotropic. Neurons die when they're overstimulated in association with large amounts of glutamate, which is called excitotoxicity. Aspartate, another amino acid transmitter, is excitatory at many synapses, while it can be inhibitory at other synapses. GABA then is widely distributed amino acid transmitter that is the main inhibitory transmitter in mammalian nervous systems. There's different types of GABA receptors. There's either GABA A, GABA B, GABA C, and these are all subscripted. Each of the different receptors have different properties and structures. GABA A is ionotropic, so that means it produces fast inhibitory postsynaptic potentials. It is made up of 5 protein subunits surrounding a chloride ion channel that can be widened or narrow. GABA B is metabotropic, and it has slow inhibitory postsynaptic potential because of the fact that it's metabotropic. And GABA C is ionotropic with a chloride ion channel. It's different from the other subunits in structure, which distinguishes itself from GABA A. Last one that is discussed for the amino acid transmitters is glycine, which is often inhibitory, while GABA is the main inhibitory transmitter. Next section is acetylcholine was the first neurotransmitter to be identified. All cells that use acetylcholine are described as having cholinergic transmission. Cholinergic receptors are important in memory and learning. So, but there's different types of cholinergic receptors. They can either be nicotinic or muscarinic. Nicotinic respond to nicotine and acetylcholine. They're inotropic, fast, excitatory, and the muscles use them. Muscarinics respond to muscarine and acetylcholine. These ones, in opposition to nicotinic, are metabotropics. They use a G protein coupled receptor. And because they're metabotropic, they're slow, and they can either be excitatory or inhibitory. There are the next section is 5 monoamines act as neurotransmitters. These monoamines can either be catecholamines or indolamines, and these two differ based on their structures. Catecholamines are either dopamine or norepinephrine. They're derived from tyrosine with a 6-sided catechol ring with molecular structure, while indolamines can either be serotonin or melatonin, and they're from tryptophan, which is a 5-sided indole ring. The next section is dopamine, which is also one of the monoamine neurotransmitters. It is found in the midbrain, the substantia nigra especially, and the basal forebrain. The different dopamine receptors are either D1, D2, D3, or D4 or D5. They're indicated in the mesostriatal pathway a lot, which is a set of dopaminergic axons arising from the midbrain and innervating the basal ganglia, including those from the substantia nigra. That's why they're very important. They mostly innervate the striatum, which is the caudate nucleus in the putamen together, which is part of the basal ganglia, and they're indicated a lot in motor control. The next pathway that dopamine is important in is the mesolimbocortical pathway, which is a set of dopaminergic axons arising from the midbrain, which in the ventral tegmental area, and innervating the limbic system and cortex. The ventral tegmental area is a portion of the midbrain that projects dopaminergic fibers to the nucleus accumbens, and they're through the D2 receptors are important in reward and reinforcement. The next section is norepinephrine. Norepinephrine or noradrenaline is one of those monoamine neurotransmitters. They're produced and released by sympathetic postganglionic neurons to accelerate organ activity, and they're also produced in the brainstem and projections through the brain. Important features of the brain that produce the norepinephrine are the locus coeruleus, which is the blue spot, and it is a small nucleus in the brainstem. It is mostly in the pons and the lateral tegmental area, and these are the two main clusters of neurons that release this. Noradrenergic is just a system that uses norepinephrine as a transmitter. The way that locus coeruleus affects the rest of the brain is that it produces norepinephrine to affect the cerebral cortex, limbic system, thalamus, cerebellum, and the spinal cord. The central nervous system has 4 subtypes of norepinephrine receptors. These can be either alpha 1 minus, alpha 2 minus, beta 1 minus, or beta 2 minus. And all of these are metabotropic receptors, so they're pretty slow. Lastly, norepinephrine is important in mood, arousal, and sexual behavior. The next section is serotonin, which is again one of those monoamine neurotransmitters. It is also known as 5-HT. It is a synaptic transmitter that is produced in the RAF nuclei and is active in structures throughout the cerebral hemisphere, and all but one of them are metabotropics. They're pretty slow acting. Serotonin mostly controls sleep states, mood, sexual behavior, anxiety, and behaviors as such. Serotonergic transmission are just neurons that use serotonin as their synaptic transmitter. These RAF nuclei where serotonin is produced are a string of nuclei in the midline of the midbrain and brainstem that contain most of the serotonergic neurotransmitters of the brain. Next section is many peptides function as neurotransmitters. The first one that is discussed is opioid peptides, which are a type of endogenous peptide that mimics the effects of morphine in binding the opioid to opioid receptors and producing marked analgesia and reward, analgesia being pain relief. The main sections that use it are the metencephalon, luencephalon, the endorphin, and dynorphin. Some other peptides that function as neurotransmitters are substance P, CCK, VIP, neurotensin, NPY, and they're in the periphery of the organs of the gut. And many are made by neurons in the spinal cord and the brain that and they also participate in synaptic transmission. Another set are the oxytocin and vasopressin, and they're mostly pituitary hormones. Lastly, some neurotransmitters can be gases. These neurons that use gas molecules, these gas molecules are able to dissolve in H2O to communicate information. One of these being nitric oxide, and also discussed earlier, carbon monoxide. Nitric oxide is more known for its functions, so it's discussed in more length. And it is a soluble gas that serves as a retrograde gas neurotransmitter in the nervous system. It is produced in cellular locations instead of being released in vesicles. It just diffuses out of the neuron as it's produced, and it diffuses into the target cell and stimulates the production of second messengers, and it can be a retrograde transmitter. And nitric oxide is mostly involved in hair growth and penile erection. A retrograde transmitter then is a neurotransmitter that diffuses from the postsynaptic neuron back to the presynaptic neuron. And that is pages 95 to 103 in Chapter 4